Thank you. My name is Rajarshi Mandal, and today I'm going to present our Multi-Omics Insights. First, we'll start with a quick introduction. One of the most enduring explanations for aging is the free radical theory, which proposes that reactive oxygen species, or ROS, generated during normal mitochondrial respiration gradually damage DNA, proteins, and lipids. Over time, that's oxidative stress overwhelms the cell's repair mechanisms, leading to cumulative damage and functional decline. Mitochondria, being both the main producers and targets of ROS, become increasingly dysfunctional with age. This leads to the polymerase gamma theory, where replication errors by mitochondrial DNA polymerase gamma amplify existing mutations. As faulty polymerase gamma continues replicating this damaged DNA, the resulting mutations accumulate clonally, impairing oxidative phosphorylation and creating a vicious cycle that accelerates aging at the cellular level. Epigenetic mechanisms, such as histone modification and non-coding RNAs, regulate gene activity during stress and aging. These alter the compactness of chromatin, controlling which genes are accessible for transcription. As fibroblasts age, these histone marks often shift, silencing repair pathways while activating stress response genes. Meanwhile, non-coding RNAs, such as microRNAs and long non-coding RNAs, adjust cell behavior by degrading mRNA and guiding specific chromatin remodelers to loci. We examine how epigenetic shifts and cytokine signaling interact to drive fibroblast aging and senescence. DNA methylation controls gene activity without altering DNA and is one of the most studied epigenetic markers. It typically occurs at CPG sites, where cytosine is followed by a guanine, and the addition of a methyl group can silence gene activity. As we age, our cells accumulate changes in methylation patterns. Some become inappropriately turned off, while others become mistakenly activated. These shifts form predictable aging signatures used to build epigenetic clocks that estimate biological age. Multiple methylation clocks were used to validate fibroblast aging and explore how mitochondrial dysfunction and treatments like dexamethasone alter the methylation landscape. The Hallmarks of Aging framework outlines biological processes like genomic instability, epigenetic change, and mitochondrial dysfunction that drive aging. This diagram maps various treatments and data sets from the Multi-Omics Fibroblast study to each hallmark. My research draws from the same data set and focuses especially on cellular senescence and altered intercellular communication. By analyzing this, we're able to probe how interconnected hallmarks evolve with age, particularly under stressors like the dexamethasone treatment and SIRF1 mutation. This systems level view allows us to contextualize cellular aging within a broader biological framework. The SIRF1 mutation encodes a protein critical for assembling cytochrome C oxidase, or complex 4, a core component of the mitochondrial electron transport chain. Mutations in SIRF1 disrupt this process, leading to COX deficiency and impaired oxidative phosphorylation, which limit the cell's ability to efficiently produce ATP. This dysfunction, seen in Leigh syndrome, provides a model for studying energy failure and accelerated aging. In our study, fibroblasts with the SIRF1 mutation displayed reduced proliferative capacity, altered bioenergetics, and early senescence, highlighting the central role of mitochondria in regulating cellular lifespan. Next, we'll do a short literature review. This study conducted a multi-omics longitudinal analysis of aging in humans. Most molecular and microbial changes occur in waves, not gradually. Panel D uses Spearman correlation to identify age-associated molecules, while Panel E reveals that different sets of molecules become dysregulated as we age, compared to a 25 to 40-year-old baseline. Importantly, as Panel F shows, only a small subset of features change consistently across age, a finding that challenges the notion of gradual aging. This reinforces the need for high-resolution datasets to track cellular aging dynamics. This figure from the same study dives deeper into how aging shapes molecular systems by using principal component analysis and unsupervised clustering. In Panel A, the first principal component from each omics dataset is correlated with chronological age, revealing that different biological layers, like proteomics and metabolics, each show unique trajectories. Panel C groups these features into 3 distinct molecular clusters. These clusters include a mixture of omics types. Aging emerges as a coordinated phenomenon involving multiple pathways and cell types, and supports our approach for using multi-omics to track aging biomarkers. This paper outlines the vital role of mitochondrial dysfunction in aging and age-related diseases, highlighting how accumulated mitochondrial dysfunction mutations and ROS production inhibit ATP synthesis and respiratory chain function over time. As shown here, the balance for mitochondrial fusion and fission is critical for quality control. With age, its dysregulation impairs mitochondrial turnover and disrupts cellular function. These amplify oxidative stress and disrupt both metabolism and cellular homeostasis. Mitochondrial decline is linked to neurodegeneration and disrupts neurodegeneration, cardiovascular disease, and cancer, making mitochondria central to aging. In our study, we observed parallel bioenergetic shifts in fibroblasts, particularly in SIRF1 mutant cells, which show impaired oxidative phosphorylation and an increased reliance on glycolysis. This study reinforces the value of DNA methylation-based epigenetic clocks as powerful tools for tracking biological age and disease risk. This figure summarizes conditions where age acceleration is observed, especially in accessible tissues like blood. Notably, age acceleration is consistently linked to a range of diseases, including Alzheimer's and obesity. This reveals altered dynamics in both SIRF1 mutant and dexamethasone cell lines and suggests epigenetic drift signals across tissues. Beyond the hallmarks we've already highlighted, our multi-omics dataset offers a rich view of aging by integrating transcriptomics and environmental stress responses. It includes over 40 fibroblast cell lines sampled across the entire replicative lifespan. This allows us to track longitudinal molecular shifts under both baseline and perturbed conditions. In addition to SIRF1 mutations and dexamethasone, cells were also exposed to hypoxia, nutrient depletion, and metabolic inhibitors, offering a wide lens on how environmental factors can also impact aging. This depth and diversity makes it uniquely powerful for cross-modal analysis, and this is why we chose it for our methodology. The full sample set was initially downloaded from the Columbia PyCard data portal. Most columns were named for clarity and conciseness, and the independent variables were separated from the original dataset. The dependent variables or biomarker measurements were sorted into 8 categories described earlier. For each of the categories, we removed columns that were not compatible with statistical tests. This included string data and identifiers. Next, we visualized some biomarker measurement distributions in each category and found that they were mostly parametric. After that, we visualized the correlations in the columns between each category. Here, we can see strong positive correlations between bioenergetics values and weak positive correlations between cytokine measurements. We identified treatments affecting aging by running students' t-tests on paired samples and the number of days grown after treatment. For each of the biomarker categories, the following statistical tests were run. To deduce which biomarkers were correlated with age, we ran Spearman's rank correlation between the biomarkers and total days grown. Next, we ran students' t-tests between the biomarkers and whether the fibroblasts had the SIRF1 mutation to determine which biomarkers were impacted by mitochondrial dysfunction. Finally, we ran students' t-tests again between the number of population doublings and Horvath's skin tube blood clot to determine whether the specific treatment had been used to figure out which biomarkers were impacted by each treatment. Next, here are our data tables for cytology, DNA methylation in clocks, bioenergetics, and cytokines. This data strongly supports the Hayflick limit, showing that cells divide less frequently as we age and stop proliferating. We see this in the declining doubling rate and divisions per passage over time. SIRF1 mutant fibroblasts, which model Leigh syndrome, had even fewer divisions due to impaired mitochondrial dysfunction slowing the cell cycle. This gives a plausible explanation for weight faltering in Leigh syndrome patients, where tissue growth is energy limited. As expected, dexamethasone further worsened these metrics by inducing oxidative stress. Interestingly, SIRF1 cells also became larger, likely due to osmotic swelling from dysfunctional mitochondria failing to regulate ion balance. Our DNA methylation analysis confirms that fibroblasts age predictably in culture, with clocks like Hannum and phenoage increasing with time. Intriguingly, Grimm age and phenoage were lower in SIRF1 mutant cells due to premature cell death. This reflects the shortened lifespan seen in mitochondrial disorders. Dexamethasone disrupted multiple clocks, especially Horvath and Hanna, suggesting that glucocorticoid treatment leaves a strong epigenetic footprint. Therefore, the normalized differential methylation index may help evaluate drug effects. Finally, deeper methylation markers like PAI1, adrenomedulin, and beta-2 microbulin revealed mechanistic links between aging and ailments such as thrombosis and kidney failure. This shows how biological age diverges from chronological age under stress. As fibroblasts age, they shift towards increased glycolysis and oxidative phosphorylation, likely due to meeting the energy demands of damaged systems. Declining mitochondrial coupling efficiency signifies greater proton leak in ROS, which supports the free radical theory discussed earlier. Cells with the SIRF1 mutation also showed a metabolic shift toward glycolysis due to inefficient oxidative phosphorylation. This cytokine data gives powerful insight into age-related immune decline and disease risk. Interleukin alpha increased with age, supporting links between inflammation and cardiovascular issues. We also saw a decline in tissue factor pathway inhibitor, explaining why older adults face higher risks of coronary artery disease and stroke due to impaired cholesterol handling and blot regulation. This explains the higher infection susceptibility and cancer prevalence in elderly. Lastly, the SIRF1 mutation increased chemokine ligand 7, suggesting that reactive oxygen species intensifies immune signaling through mitochondrial dysfunction. Our study offers clear biological evidence for several foundational theories of aging. The SIRF1 mutant fibroblasts exhibited elevated ROS, impaired oxidative phosphorylation, and increased glycolysis, supporting the free radical theory of aging. We also observed age-related cytokine shifts, including reduced PCSK9 and elevated interleukin, which are linked to immune dysfunction and neuroinflammation, offering an explanation for the increased risk of Alzheimer's and cancer in the elderly. Furthermore, bioenergetic profiling revealed reduced ATP production and mitophagy, confirming mitochondrial dysfunction as a driver of senescence. Lastly, several epigenetic clocks deviated from expected trajectories under stressors like dexamethasone, indicating that these responses may accelerate biological aging beyond what is seen with time alone. In the next phase of our research, we developed a machine learning framework to predict biological age from DNA methylation with a large data set of over 480,000 samples across 6,000 humans. We begin with ridge regression-based feature selection to identify the most age-informative CPG sites, then trained 20 machine learning models and evaluated performance with bootstrap confidence intervals. The top 4 models were further analyzed for model interpretability using SHAP and attention-based methods. We then constructed a weighted ensemble to improve accuracy and applied it across 9 age-related diseases. This allowed us to explore epigenetic age acceleration in diseases like Alzheimer's and Parkinson's, offering both predictive insights and biological relevance. Our goal is to move towards clinically trustworthy, interpretable epigenetic clocks that deepen our understanding of how disease and aging intersect at the molecular level. Thank you for listening.
Thank you for the talk. Do you have any questions? We have time for a really quick one. In your opinion, which would be the data type that fits better with aging? Because you mentioned many different data sets. Which one do you think is the most promising for the machine learning approach?
I think that either methylation values or cytokine are most important for machine learning because they can be easily measured multiple times across the lifespan of fibroblasts and therefore yield the most information for machine learning models.
Thank you very much.